Good afternoon, dear colleagues and guests. Today it is the 16th of May 2022, 12 p.m. according to Moscow time. Allow me to consider open the meeting of the Dissertation Council for the defense of Asya Davidian's dissertation for the degree of the candidate of biological sciences in the scientific specialization 1523, Developmental Biology, Embryology, on the topic Functioning of the Nucleolus Organizer Region in the Growing Oocytes of Sauropsida. According to the order of Alexander Gnetov, St. Petersburg Scientific Secretary, dated from the 21st of February 2022, number 1410/me, Elena Yermilova, Doctor of Biological Sciences, Professor of the Department of Microbiology, St. Petersburg University, was appointed the chairperson of this Dissertation Council, and by the same order, the members of the Dissertation Council were approved. Let me introduce them. Anna Malashecheva, Doctor of Biological Sciences, Associate Professor of the Department of Embryology, St. Petersburg University. Alexander Rodionov, Doctor of Biological Sciences, Professor of the Department of Cytology and Histology, St. Petersburg University. Dmitry Bogolyubov, Doctor of Biological Sciences, Chief Scientific Officer, Head of the Laboratory of Cell Morphology, Institute of Cytology, the Russian Academy of Sciences. In the remote interactive mode, we have got Jana Bridger, PhD, Director of the Center for Genome Engineering and Maintenance, Brunel University, London, the United Kingdom. Can you see and hear us?
Yes, I can.
The answer is positive. Our applicant, Asya Davidian, is also here with us. Also, there is a scientific supervisor, Doctor of Biological Sciences, Professor of the Department of Cytology and Histology, St. Petersburg University, Elena Gaginskaya. To improve the quality of communication, I ask you, dear colleagues, to turn in the remote interactive mode, turn off your microphones, but do not forget to turn them on when you are given the floor. I also inform you that audio and video recording of our meeting is being conducted, as well as live broadcast on St. Petersburg University website. Simultaneous interpretation of speeches from Russian into English and from English into Russian is being provided. At this time, an email address is posted on live broadcast of the meeting of the Dissertation Council, where during the meeting all listeners can express their opinions and set questions to the applicant online concerning her dissertation and the current scientific discussion of her speech. These questions will be forwarded to me by the technical services, and I will voice them during the discussion. Questions must be related to the applicant's presentation and the content of the dissertation, and the name, position, and place of work of the author of the question must be indicated. Questions that are not related to the scientific discussion, to the discussion of the dissertation, to the text and assessment of the dissertation itself, and anonymous questions will not be voiced. In accordance with the procedure for awarding the academic degree of Candidate of Sciences, academic degree of Doctor of Sciences at St. Petersburg University, approved by local regulations of St. Petersburg University, hereinafter the procedure, a meeting of the Dissertation Council is considered eligible if at least 2/3 of the approved membership of the Council takes part in its work, but not less than 4 people. Our Dissertation Council consists of 5 people. 5 members are present, including 3 members in the remote interactive mode. All members of the Dissertation Council and the applicants are provided with audio-visual contact. Thus, there is a quorum. I ask an employee of the department for supporting the activities of Dissertation Council, the curator of the defense, to issue an attendance sheet. The procedure of today's meeting of the Dissertation Council, with a total duration of approximately 2 hours, is the following. Number 1, a brief report of the chairperson on the documents submitted by the applicants for a scientific degree and their compliance with the established requirements. Answers to possible questions, about 5 minutes. A short presentation by the applicant for a scientific degree reflecting the main points of her research, approximately 15 minutes. Questions to the applicant, strictly according to her report, no more than 2 minutes per question. Answers of the applicant, no more than 5 minutes for all questions. Speeches of all members of the external reviews, if they are received. Speeches of all members of the Dissertation Council, in turn, with the assessment of the dissertation research, with a summary of their positions, with questions and suggestions for the dissertation, about 10 minutes per speaker. The speech of the chairperson with the review, about 10 minutes. Answers of the applicant for a scientific degree to questions and comments of the members of the Dissertation Council, no more than 20 minutes. Open discussion. Speeches at the request of those present at the defense of the dissertation, with a summary of their positions and or with specific questions and proposals for the dissertation, strictly on the topic of the research, no more than 2 minutes for everyone. At the same time, all interested people are requested to register in the registration form and completely introduce themselves before your performance. You should state your name, position, place of work. Presentation by the chairperson of questions addressed to the applicant, which came in order to discuss her speech and scientific discussion on the broadcast of the meeting of the Dissertation Council on St. Petersburg University website. I would like to draw your attention to the fact that written questions which take more than 2 minutes won't be voiced. Answers of the applicant, no more than 2 minutes per question. Speech of the scientific supervisor of the applicant, no more than 3 minutes. Discussion of the results of the defense by the members of the Dissertation Council before the open vote, during which the sound of the broadcast is muted. Approximately 5 minutes. Open roll call, counting of votes by the chairman of the Dissertation Council with the entry of the results into the minutes of the meeting. Making a decision on awarding or not awarding an academic degree. Concluding remarks of the applicant, no more than 2 minutes. Dear colleagues, are there any questions or objections to this regulation? If there are no objections, I will proceed to the implementation of the rules of the meeting, but before, please turn off the sound of your mobile phones. Please, dear colleague, in the remote interactive mode, please, Jana, don't switch off your phone in order to communicate with you quickly in case of technical breakdown. Thank you. So I'm starting to conduct the meeting. The dissertation of Asya Davidian for the degree of the candidate of biological sciences, specialization 1523, developmental biology, embryology, on the topic functioning of the nucleolus organizer region in the growing oocytes of Sauropsida was accepted for the defense by the order of the Academic Secretary of St. Petersburg University from December 28, 2021. number 13036/1, Asya Davidian wrote her dissertation research on the basis of St. Petersburg University. Her scientific supervisor is Doctor of Biological Sciences, Professor of the Department of Cytology and Histology, Yelena Gaginskaya. The number of publications of the applicant in which the main scientific results of the dissertation are stated, according to the list presented, 4 publications. They are published in journals indexed in the scientometric databases Web of Science and Scopus. All the documents submitted by the applicant on the information I received from the curator meet the requirements and are stored in the applicant certification file. Their copies are held by an employee of the Department for Maintenance the Activities of Dissertation Councils, who is now in the hall. Dear board members, do you have any general questions to the applicant, and if there is a necessity to announce the entire list of documents submitted by the applicant for the defense? No questions. And now I would like to give the floor to our candidate, Asya Davidian. Thank you very much. Distinguished chairperson, dear board members, dear colleagues, I would like to bring to your attention a report that outlines the main results of my dissertation. The nucleolus organizer, NOR, is the most important region of the eukaryotic genome, which contains multiply repeated clusters of ribosomal genes separated by IGS intergenic spaces that are variable in length and composition. The activity of NOR on the chromosome leads to the formation of the nucleolus, the main function of which is the biogenesis of ribosomes. The formation of the ribosome requires the presence of 4 RNA molecules in an equimolar ratio. 3 of them are encoded in rDNA. NOR repeats and are read by RNA polymerase 1 as a single RNA precursor after reassembled in the granular component of the nucleolus with the participation of 5S RNA and ribosomal proteins. As for 5S RNA genes, it is believed that in most higher eukaryotes and in particular in vertebrates, they are organized into separate clusters that are not associated with NOR and are transcribed by another enzyme, RNA polymerase 3. The relevance of studying the features of the functioning of NOR in oocytes is determined by the fact that during the growth of the oocytes, along with the other RNAs, huge amounts of ribosomal RNAs are synthesized, are stored. In many cases, chromosomes are transformed into transcriptionally active so-called lamb brushes, and amplified extrachromosomal nucleoli can participate in synthesis of ribosomal RNA. This maternal RNA reserves an essential to ensure early embryogenesis prior to the activation of the zygotic genome, and possible ways to provide oocytes with the necessary amount of rRNA were clearly summarized by McGregor in 1982. These are rDNA amplification in the oocyte's nucleus, supply of ribosomal subunits from auxiliary cells. The slide shows options for organizing the embryonic vesicle using the example of amphibians, reptiles and birds. In turtles, apparently, as well as in amphibians, amplifications of the rDNA of the nucleolar organizer occurs in the oocyte nucleus, resulting in the formation of many extrachromosomal nucleoli. At the same time, scaly reptiles and birds demonstrate the absence of nucleolus at the stage of developed lamb brushes. Although, according to modern phylogenetic analysis of Sauropsida clade, squamates are the most distant group in relation to a Achalazoa group, which unites birds, crocodiles, and turtles. This determined the choice of objects of work. The aim of my work was a comparative analysis of the structure and functioning of NOR in growing oocytes of Sauropsida representatives using the example of Acleosaura group, which includes birds, crocodiles, and turtles. The study was conducted on model animals such as a domestic chicken, Gallus domesticus, and the red-eared slided turtle, to study the morphology of the ovary and the cytomolecular features of the functioning of ribosomal genes in developing oocytes in immature and mature female domestic chickens. To study the cytomolecular features of the functioning of ribosomal genes in developing oocytes in red-eared turtle. To study the in silica the structure of the ribosome repeats in the genome of the red-eared turtle, and so on. In the chicken, the only NRR is located in the genome on chromosomes 16. In growing bird oocytes, the chromosomes pass through the typical lamb-brush stage. However, in the ovaries of mature chickens, nucleoli in oocytes have never been detected, whereas in about 40% of early diploid chicken oocytes, 1 or 2 nucleoli were found, which disappear at the lamb-brush stage. To explain the phenomenon of inactivated NRR in oocytes of mature birds, a hypothesis was put forward about the existence in the juvenile ovary of 2 morphological forms of oocytes of the early diploid stage, which differ from each other by the presence or absence of a nucleolus in the nucleus. At the same time, oocytes without a nucleolus presumably remain in a dormant state until the onset of puberty of the female. The first task was to test this concept. First, oocytes were analyzed in the ovaries of chickens shortly after hatching on serial paraffin sections stained with hematoxylin eosin. After obtaining a series of images of sections of the ovary, we aligned them with each other, followed by a 3D reconstruction of the entire ovary in the AMIRA program. In addition, 3D reconstruction revealed anterior, posterior, and dorsoventral polarity in oocyte maturation. As a result of the analysis of chicken oocytes of different ages using RNA fish with a probe to the internal transcribed spacer, as well as using immunohistochemistry with antibodies against the nucleolar marker protein, fibrillary, we showed that the nucleolus appears and functions at the early diplantine stage. The nucleolus functions at an early stage of lamb brushes, after which an early middle stage of lamb brushes, the NRR apparently becomes inactivated and the nucleolus degrades. The nucleolus first disintegrates into large fragments containing pre-RNA and proteins, and then into small granules in which fibrillary is detected and does not contain pre-rRNA. Moreover, when studying the ovary of a sexually mature chicken, we found for the first time a functioning nucleolus in early diploid oocyte. In oocytes at the lamb brush stage, the nucleolus disappears. Thus, it has been shown for the first time that an identical scenario of NRR functioning is observed in oocyte in chickens and mature hens. In the red-eared turtle, the only NRR is located on microchromosome 14. In the germinal vesicle isolated at the same stage of oocyte growth as in the chicken when stained with Sitax green. In addition to lamb brushes, many extra chromosomal bodies are observed in the nucleus, some of which are probably amplified nucleoli. To verify this, the isolated oocyte nucleus was stained with antibodies against the nucleolar marker protein, fibrillary. But how do these multiple nucleoli arise? Do they appear all at once, or does their number gradually increase with the growth of the oocyte? For this purpose, the RNA fish method with a probe to an external transcript spacer was used on serious sections of the total ovary to detect immature RNA transcript. We have shown that early diploid total oocyte contain 1 to 2, sometimes 3 nucleoli. With the growth of the oocyte, the number of nucleoli increases. In the germinal vesicle with developed lambrushes, the number of nucleoli reaches several hundred. To answer the question about the mechanism of the increase in the number of nucleoli with oocyte growth and whether our DNA undergoes replication in the nucleolus, we undertook immunostaining with antibodies against the pCNA replication marker. As a result, we have shown the fluorescence of nucleoli in oocytes from early diplotin to the stage of developed lambrushes. Apparently, our DNA amplifications in the nucleoli continues throughout the entire period of total oocyte growth. According to the literature and our data, we shown on the phylogenetic tree, among which vertebrates NRR amplification in oocytes is described. These are ray-finned fish, amphibians, turtles, and crocodiles. Turtles and crocodiles differ in the DNA amplification strategy from the studied fish and amphibians. It is known that meiotic extrasynthesis of RDNA in the latter occurs in the Pachitin stage and leads to the formation of a cap. As mentioned earlier, the ribosome contains 4 rRNA molecules in equimolar ratios. We have shown that high-level NRR amplification occurs in the oocyte nucleus in turtles. The question arises of maintaining a quantitative balance between the 18S, 5.8S, and 28S rRNA genes that are parts of amplified nucleoli and the 5S RNA gene for creating ribosomes. When analyzes the structure of the tuberculate total ribosomal repeat, we were surprised to find an insertion of the 5S RNA gene into the IGS intergenic spacer. This gene is directed in the opposite direction with respect to the DNA cluster. This NR5S RNA gene exists along with a separate cluster of canonical 5S RNA genes, which as in all vertebrates lie outside the NOR. The difference between the NR5S sequence and the 5S rRNA gene within the same species is approximately 20%, which makes it possible to distinguish them using PCR. It was necessary to check whether NR5S rRNA gene is functional. It was necessary to check whether NR5S rRNA fits into a functional molecule capable of entering the ribosome. It was necessary to verify the presence of NR5S transcripts in various total tissues and check if NR5S rRNA is a part of functioning ribosomes. When analyzing the secondary structures, we see that NR5S rRNA fits into the secondary structure characteristic of functional 5S molecule, which can enter into the large subunit of the ribosome. To test for the presence of NR5S transcripts in total tissues, we analyze the transcriptomes of the ovary, testes, and somatic tissues of the Chinese. 3 kill total available at the NCBI. We confirmed the content of NR5S transcripts only in the ovary by isolating RNA from various tissues of the rat ear turtle, including the ovary, heart, skeletal muscle, and testes. Moreover, by separating the oocyte from the follicular epithelium, we could be suspected of supplying the oocyte with ribosomes, we show that an NRF5S rRNA is specifically synthesized in amplified oocyte nucleoli rather than in follicular cells. And the last task was to clarify whether NRF5S rRNA is a part of functioning ribosomes. For these lysates from oocytes were ultracentrifuged in a linear sucrose gradient in order to obtain monosomes and polysomes. After RNA isolation from each fraction, we assess the representation of various ribosomal RNAs in the fractions using quantitative PCR or cDNA templates with primers to NRF5S, canonical 5S, and 28S rRNA. In the experiment with oocytes about 5 millimeters in size, 5S rRNA and NRF5S show the same representation pattern in all fractions, which apparently indicates their presence in functioning ribosomes. In the experiment with vitellogenic oocyte about 10 millimeters, canonical 5S rRNA is found mainly in the two upper fractions, while NR5S rRNA is predominantly in monosomic and polysomic fractions. Apparently, during oocyte growth, canonical 5S rRNA is replaced in ribosomes by NR5S rRNA. Thus, the fact that the functional 5S rRNA gene is included in the NR in totals may indicate that the balance between the 18S, 5.8S, 28S, and 5S rRNA genes is maintained during oogenesis with RDNA amplifications. We also reviewed available NCBI data on ribosomal repeats in vertebrates and found a similar insertion of NR5S in IJS only in turtles and crocodilians. Despite the intergenic spaces differing greatly in length and composition, we see that no NOR5S insert extremely conservative. Since we did not find the NOR5S insert in ray-finned fish, amphibians, and scaly reptiles, we assume that such an insert arose in Achelozoas in the Permian and probably disappeared the second time in the ancestors of birds, since we did not find traces of it in the IJS in bird representatives. Conclusions. To save the time, allow me not to read out conclusions. 3 articles were published, and thank you very much for your attention, and don't hesitate to ask questions. Asya, thank you very much to your report. I have a question to board members. Do you have any questions to our applicants, strictly according to the report, reflecting the main provisions of the research, no more than 2 minutes per question? I have got questions concerning what is the composition of amplified ribosomal genes in amphibians and turtles. Are they circular or linear molecules? Amphibians have ring molecules, but in electronic microscope we found out linear molecules, but it is an artifact when DNA appears. These are fragments of ring molecules. They are replicated with the help of rolling ring. One more question. And the gene you found out, the fifth in the composition of 45th gene, they are different by 20 nucleotide from canonical. It is a big figure in order to preserve secondary structure. Are there compensational substitutes? We paid attention to Let me find the slide. We paid attention to those curves and spirals, which are very important for entering ribosome composition. And we found out that curve C and third spiral, they are conservative, and they are it is responsible for ribosomical unit. And under certain conditions, 37 degree, it is part of secondary structure, and there are certain variations. They are shown in the slides. Red color in most conservative places, black line color. They are conservative in all eukaryotes, and they play a big role, an important role for secondary structure 5S and her functionality in ribosome, and for entering to ribosome, spiral and curve 3, they preserved their composition, but there are substitutes, loop B, but I'm not sure how it is important for connection with proteins in ribosome. These substitutes should be necessary for preservation of secondary molecule. If T is changed by G, then C appears in complementary position. It's very difficult to assess. Dear board members, do you have any other questions? If there are no questions, let us turn to the speeches of board members with their reviews and evaluation of the dissertation research, with questions and suggestions to the applicants, and external reviews and remarks and negative assessments. I remind you that our applicants can answer after each speaker or after all speeches. I offer you to answer after each speaker to the questions or remarks. Do you have any objections? No objections. If there are no objections, I would like to note that all reviews were published at St. Petersburg University website. To read them in advance, I offer you to focus only on the main points, questions and remarks to the applicants. Do you have any objections? No objections. Now I would like to give the floor to Anna Malashicheva. I don't have any remarks in my review. I would like to focus on the main moments. The work is brilliant, and the classical evolutional approach was used at the level of morphological analysis, and the work is unique when classical methods are combined with ultra-modern methods, and our candidate is modern, qualified university biologist. Chapter conclusion is devoted to comparative evolutional analysis of functional of ribosomical genes in vertebrates, and generalization of the research is shown. And this section proves scientific and professional maturity of our candidate. The conclusions are clearly formulated and they are adequate. On the whole, the work is completed at very high international level, and it is original, and the data are relevant. Thank you very much, Anna Borisovna. If there are no remarks, now we give the floor to Alexander Vikentьевич. It was a great pleasure for me to be the board member. This work, I remember, when 45 or 47 years ago, the first postgraduate students studied this problem. How if there is a nucleolus in oocytes of mature chickens, and 50 years later, technical opportunities have radically changed, and it was very interesting to investigate this problem, and the result is very interesting, and it was in time and in place, and we see as a result of the study, principal and new observations were presented, and genes 45S work in mature birds. I'm not going to focus. I'm not going to focus in detail in literature review materials and methods of this study. They are written at high professional level. I have two small materials and methods are very important aspects of the work if we speak about dissertation, PhD dissertation, because all in all it proves the level and preparation of the researcher for further work. It is an important element of qualification paper. And the main section of the work, it is devoted to the peculiarities of organization of genes of 45S rRNA in genome and in oocytes in birds and Sauropsida, crocodiles and turtles. It is these results are very interesting. Mature birds in a part of cells, 45 genes sRNA continue working. Bioinformatics is taken into account. It was shown that there are special sequences coding 5S ribosomal RNA, which are situated in the cluster of ribosomal genes. We knew some objects were shown. Then on chromosomes 5S genes, they are close to 45S genes RNA. Plants have such several cases, and here it is situated in transcribed units, and whether this gene is transcribed, inserted into ribosomal genes of I would like to highlight this part of work is interesting that it is considered in broad evolutional aspect and concerns broad spectrum of questions connected with the organization and evolution of ribosomal genes in this complicated and phylogenetic evolutional group as Sauropsida. It attracts a lot of attention to this study. And my main remark concerns the pluses and the advantages of the work. 5S genes are within the limits of clusters of 5S. The hypothesis that it is the form recapitulation, the appearance of a peron, in functional sense connected with amplification in order to level the genes, the count of amplification. The idea is original. My main remark concerns one intermediary conclusion connected with the mechanism of amplification of ribosomal gene in genesis. How do these amplified copies of ribosomal genes in oocytes appear? 3 ways. At the count of DNA RNA-dependent RNA polymerization, and the role of amplifications of chromosomal copies. Chromosomal copies of these amplified genes, as it is offered by our candidate, it can be constant amplification of already amplified fragments. And how can we choose between these variants? The work uses nuclear antigen, and candidate shows that this amplified nucleolus produce strong signal, and the peculiarities of binding these agents with other proteins. They are not considered only one reference to the work of 1993, general reference. It is important to know here that after amplification occurred, PCNA does it quit matrix? Can we see it in the replication, or it can be preserved on replified copies? Doesn't continue living there. These proteins can be bound with different proteins in the cell, with elements of nuclear matrix, and with some other proteins. These questions was discussed before, and it seems to me that it would be better to consider in detail at the modern level the peculiarity, the question about to what extent or how long are these antigens PCNA found out only in replication area or in other structures. It is the main question. As for constant replication of the amplified genes, there are a lot of problems. Ring molecule, this should be certain balls after multiple amplification of ring molecule. It is very tricky structure. On the whole, I liked the work. A professional level methods, high quality of photos represented, and the conclusions are without doubt. And it seems to me that the dissertation conforms with the requirements stated by for the dissertations at St. Petersburg University. Alexander, thank you very much. Asya, please answer the questions and remarks. Alexander, thank you very much for such a detailed analysis of my work and important comments. Regarding the issue of PCNA, I can say the following thing. This protein belongs to the so-called sliding clamp proteins, which assemble into multimetric structures surrounding the DNA ring and the replication fork region when DNA polymerase adds nucleotides to the growing strand. Reviewed by PCNA is delivered to the DNA template by replication factor C, which is also involved in the disassembly of this complex after replication is complete. In answering your question, whether pCNA remains on the daughter DNA strand indefinitely, no, pCNA is disassembled into monomers after each replication event. I am familiar with Stanislav Narishny's dissertation on the identification of the broad functional significance of pCNA, for example, on its possible participation in glucolysis processes. In confirmation of this, we also detected pCNA in the oocytes and in the cytoplasm on follicular cells on total ovary cryosections. At the same time, it is known the main function of pCNA is to ensure DNA replication and repair. This is described in detail in the review of Boehm 2016. In order to make sure that our pCNA data reflects DNA replication in the nucleolus, I set up an experiment with the inclusion of a modified thymidine analog, considering that this was the only non-repetitive experiment. I did not include the result in my dissertation. But to answer this question, I can show the incorporation of EDU into the extrachromosomal nucleoli of the oocyte at the Lamb-Brush stage. I fully agree with the rest of Alexander's remarks, and I am grateful for them. Alexander, are you satisfied with the answers of our candidate? And now I would like to give the floor to Dmitry Bogolyubov, distinguished board members, chairperson, candidate, colleagues. I'm not going to focus in detail on the actuality of this work. It is obvious from my point of view, as a result of the study, experimental research, the candidates managed to obtain new and important results, which can be interpreted as a scientific achievement, and they open new prospects for further research. And in the context of modern embryology, nuclear, and so on. In the work, our candidate demonstrated impressive diversity of the ways of formation of ribosomal RNAs, the absence of applications of genes of nucleolus organizer in chicken was proved. The character of functioning and dynamics of nucleolus organizers is not different in all sites of mature and immature birds. As for total, the high level of amplification of ribosomal DNA was demonstrated. We can see it in amphibians, but the mechanism of forming of these nucleoli is different in turtles and amphibians. In a space of RDNA of turtles and crocodiles, sequence of NOR5S was studied. And candidate proved that this sequence is evolutionarily conservative for crocodiles and turtles, and its expression is specific for oogenesis. And NOR5S RNA is a part of functional ribosome compositions. I'm not going to focus on the structure of the work. It is written in my review. A literature review is laconic, very informative and clear, very well illustrated. At the beginning, there are general data about the composition on nucleoli organizes and nucleoli. And if we speak about nucleoli composition, it will be better to, for example, complicated ultrastructure was described by the author. It is defined by secondary phase separation of polymer biomolecules, and you should have focused on these facts in more detail. And why do turtle have two component nucleoli and other reptiles have three nucleoli? Three component nucleoli. And what are the reasons for such differences? Then the compositions of ribosomic genes was considered taking into account the peculiarity of functioning. And I have a technical question. Speaking about the stages of chromosomal brushes development, you mentions the stage of compensation, postal rim-brush stage. Do you mean karyosphere stage? And why is karyosphere is a cytological phenomenon of oogenesis is not mentioned? Is cryosphere formed in turtles and radiate turtles and in other turtles, and what is it? There are data about the sources of ribosomical RNA in oogenesis and amplification of nucleolus organizer as a leading mechanism of RNA synthesis. And specification here giving transition of the function is a characteristic of ovaries of insects. They were studied on the example of Drosophila, and the egg of Drosophila is not always at the stages of 9 and 10. According to Gering's classification, there is temporary outburst, the compactization of cryosphere, and this phenomenon is specific for oogenesis of Drosophils, and it is not the best example for the activation of oocyte nucleus. Sections concerning peculiarities of functioning of nucleolus organizer of reptiles. It is stated that according to the study, only in juvenile ovaries nucleoli can be contained. As for materials and methods, this section is very impressive and proves the high professional level of the candidate. The protocols represented are without doubt, and it proves that our candidate is very professional and of high interest. Is part which is devoted to the results and the discussion. As for section 3.1, which is devoted nucleolus in chicken oocyte, two main findings according to 3D reconstruction of histological methods. 86 oocytes were found out, 86% against 40 And if I'm not mistaken, you speak about significant specification. The rest, 14% of oocytes of chicken don't contain nucleoli. Please comment upon that. As for the section devoted to detection of choline, it is without doubt. And I ask you to prove why you were interested in a nuclear distribution of choline. Secondly, speaking about the fact that the presence of choline inside the nucleoli, it can be result of natural movement of protein inside cell nucleus. It should be noted that here we speak about the dormant state. And in natural conditions of choline accumulation in nucleoli, in neurons of black substances in human brain, it is a rare phenomenon connected with the changes in physiological conditions. And please express your opinion about possible functioning meaning of such high level of choline in nucleolus in oocytes of mature mature hen. And section 3.2 is devoted to the nucleolus in oocytes of turtle. For the first time, different choline containing nuclear bodies were found out. They are flash negative, according to your opinion. There is a question: how can you explain the lack of body histonical locuses in diptatin oocyte in turtle at this stage? As for scientific novelty of this study, the next sections are very important. The results of the research of ribosomal repetition of turtles. I don't have questions concerning this part. I would like to congratulate you on an important discovery. The results of this study were tested at high-rating journal. In my opinion, this story with Noro5S RNA deserves the level of successful candidate dissertation. The conclusions of the dissertations conform with the aims and tasks of the research. I don't have any principal serious objections and remarks. I'm not going to focus on different stylistical inaccuracies and punctual mistakes, punctuational mistakes. Apart from that, all my questions, they are of scientific interest, and the remarks of recommendational character, and they don't spoil overall positive impression from the dissertation. I'm sure that Davidian's dissertation is a completed mature work, experimental research. It is novel and it is accomplished at high level, and modern methods were used, and the results are reliable. Conclusion: the dissertation of Asya Davidian is a completed scientific and qualified independent research accomplished at high methodological level, and it is based on diverse experimental material. The dissertation contains proved provisions. As for actuality, and as for relevance, novelty, scientific and practical significance, according to that, Davidian's dissertation conforms with the requirements stated by the order procedure on awarding academic degrees at St. Petersburg University. And our candidate, Asya Davidian, deserves the degree of the candidate of biological sciences, specialization, developmental biology, embryology. Points 9 and 11 were not violated by the candidate. Thank you very much. Dmitry, thank you very much. Asya, please answer the questions and remarks. Dmitry, thank you very much for such a detailed analysis of my work. All your questions and remarks seem very important and interesting to me. I'll try to answer them in order you voice them. Regarding the description of the nucleolus from the standpoint of physiochemistry and phase separation of polymeric biomacromolecules, I am familiar with these works where ideas about membraneless organelles are being developed as complexes based on the separation of liquid phases. And maybe it's my omission that I did not mention it in the literature review. And as to why the nucleoli in turtles are two components, while in other reptiles studied, active nucleoli include all three components. Indeed, in all reptiles, with an exception of turtles, the nucleolus has a three-component organization, just as in mammals and birds. At this stage, it is difficult to judge the reasons for the differences. According to Thierry and Lafontaine, it is correlated with the length of the intergenic spacer. The authors assume that an increase in the length of the spacer ensures the separation of the fibrillar components into fibrillar center and the dense fibrillar component due to the looping out of the IGS into a new compartment. And some authors, Lamayette, believe that the two-component nucleolus of turtles may indicate that they belong to the most primitive reptiles. And as for the functional significance, so far it can only be assumed that the appearance of a fibrillar center is amniotics, where DNA genes and a pool of transcription factors are concentrated, ensures a high rate of rRNA synthesis. And this fibrillar component is dissipated. As for the next question regarding the formation of the karyosphere and the oocytes of turtles and crocodiles, the chromosome condensation stage or post-Lamb-Brush stage refers to the period of oocyte development when the chromosomes are inactivated, condense, and can assemble into the karyosphere. The study of the formation of the karyosphere was not part of our task. According to the literature data, the karyosphere in the red-eared turtle is formed at an oocyte size of about 1 centimeter. Grouped bodies, presumably nucleoli, are seen in the oocyte nucleus. This work does not mention the presence of karyosphere capsule. There are no data on the karyosphere in crocodiles. regarding the fact that the transfer of the function of RNA synthesis from the oocyte to the trophocyte is characteristic of the meroistic ovaries of insects in general, including teletropic, are not only polytrophic. This concerning the remark about the transcriptional inactivity of the Drosophila oocyte nucleus. The oocyte itself exhibits synthetic activity at certain stages of the prophase or of the first division of meiosis. I also mentioned this on page 19 when I referred to the nearly transcriptionally inactive germinal vesicle. Regarding I dwelt in detail on the example of Drosophila polytrophic overall, since this is a model object and clearly demonstrates the meaning of polygenomic oogenesis, where the oocyte is nourished by germ cells. As for the interest in nuclear distribution of choline, I can say the following. Choline is the main marker protein of cagile bodies. Since the latter are associated with the functioning of the nucleolus, it was interesting to study the dynamics of choline localization in chicken oocytes. There are many works showing the close relationships between the nucleolus and the choline protein. Our detection of choline near the nucleolus and further in the decaying nucleolus in adult chicken oocytes was consistent with the work of Raska, 1990, Gilda, 2019, Broom and Herbert, 2013. They showed the role of choline in NOI inactivation in response to stress or DNA change or damage. I agree that choline may also play a role of an OR inactivation during oocyte growth. In answering to the question of how the absence of histone Loci bodies in total oocytes at the Lamb-Brush stage can be explained, as assembly of histone Loci bodies requires active transcription of histone genes. They have been described in oocytes of Drosophila, newts, and cloud frogs. At the same time, it is interesting that TGLs have not yet been detected in oocytes at the Lamb-Brush stage of domestic chicken. This is evidenced by Krasikova's article 2012, where RNA fish with BAC clone containing histone genes was performed on preparation of chicken lamb brushes. Only chromosomes were labeled, not loops, rather than loops of chromosome 1. No bodies associated with this region were also identified. We also never observed any bodies associated with certain regions of the chromosomes on lamb brush preparations from the red-eared turtle. I agree that the histone genes in turtle oocytes may be transcribed at an earlier stage of oocyte growth from which we have not isolated lamb brushes. This question, as well as the question of the nature of heterogeneous choline containing nuclear bodies, requires a special study. In response to the question of how the absence of colocalization between choline U3 and small nucleolus RNA can be explained, I would like to give similar example from the work of Joko Gell on cloud frog oocytes. The authors of this article also failed to detect, using in situ hybridizations of U3 small nucleolus RNA in choline-containing bodies, despite the fact that as a result of injection of U3 RNA into the nuclei of Xenopus oocytes after 15 minutes, their localizations in cagile bodies was shown. At the same time, within an hour after the introduction of U3 RNAs in the the intensity of the label in Cajal bodies decreased, while the intensity in the nucleoli increased. The failure to detect U3 RNAs in Cajal bodies by fish in both turtle and xenopus oocytes has not yet been explained. It can be assumed that the resolution level of these two methods is different concerning whether all chicken oocytes contain nucleoli. The odd ideas were based on the analysis of thin sections with a thickness of 2 or 4 micrometers, so that the nucleoli did not fall on all sections. Chin did not take into account the ovarian zone in the analysis of oocytes. I made thicker sections of 10 micrometers and analyzed on those nuclei that completely entered the section. We believe that the nucleolus is formed at the early diplotin stage, and these 14% of oocytes without nucleoli are classified as oocytes in which the nucleoli has not yet formed. Although I also allow the possibility of certain technical errors, for example, in determining the early diplotin stage in some oocytes without a nucleolus. With other comments regarding typos, grammatical errors, and stylistic inaccuracies, I fully agree. Asya Genrikovna, thank you very much. Dmitry Sergeyevich, are you satisfied? Yes, I'm fully satisfied. Thank you very much for detailed answers. Now I would like to give the floor to Jana Bridges.
Hello, can you hear me? Everybody can hear me? Yes, good. Okay. Thank you very much for inviting me and letting me read your thesis. I have been through it 2 times and enjoyed it very much. It is a very impressive tour de force of work, and you should be very proud. I'll go through my thoughts, and then I have a couple of remarks that perhaps you can address. So thank you for doing that. So the literature review I found really interesting, and it was very important and exciting to see the old papers and going back to old data, you know, where these sort of hypotheses and postulations and dogma were set originally. So seeing all those papers and names of colleagues fully cited, and then bringing that up to date with very modern, right up to date literature was very good. So the distribution, the way you had looked at the literature and then put that into your dissertation was exceptional, and that was a very nice piece of work to read. Also going through the methods, they were very detailed, and it's very important that people can then follow what you've done and even then recapitulate that in their own laboratories or in their own studies, and that's very important in science globally that can happen. So that was also very well written, and I read the dissertation in English, and I really had very few comments to make about the English, so I won't even bring them up. So thank you for that. What has been so impressive and extraordinary about this piece of work is that you have taken old postulations, hypotheses, and dogma, and then applied these very modern techniques in a very professional way, as my other colleagues on the committee have said. So, and then being able to find these very important findings that are not only relevant within the field of the organisms that you're looking at, but also important for then other people to take on to look at in other organisms, and ultimately for female fertility. So I think it's not just important in the area that you're working in, but it has connotations right across oogenesis in a number of organisms, and so that is to be commended. I think what is very nice about so you have these three areas, these three sort of results chapters that we go through, and you lead us very well through that in your dissertation. But what knocks it out the park is this, you have a number of findings, and we have reiterated them here a number of times now, but where you then set forward with the bioinformatics, and quite often we get a bioinformatics study, data analysis, and looking at that, but then you've gone back to the laboratory and then taken the findings that you found in silico and put them with going back to the lab and proving them in reality. And I think that takes that to another level again. So I congratulations on doing that, and doing it very, very nicely. I have two questions I hope that you don't mind addressing in this situation. Is that I'm really interested in the nucleolus and in other proteins that you find there, and it's really exciting to look at these different structures and their function in a range of organisms. And too often in science we end up looking at the same organisms that everybody else looks at, and they're not always perhaps the best organisms to look at. So I would really like to know, have you got any comments to make about other proteins that are found in the nuclei of the organisms that you looked at? So I know you use fibrillarin as a marker and a nucleolin, but do you have any knowledge that you could impart this afternoon, it's morning here, about other proteins that you could have used as markers in there? The only other comment that I would like to make, and I have no negative thoughts about your data and how solid it is and how rigorous and significant it is, but I would like to see if I can have an overview of the quantification, the numbers that you end up having to look at. You did this methodology that very, very carefully with the sectioning of the ovaries and looking at all these cellular structures and components. And I just wonder if you could talk through the kind of numbers that you assessed. You have percentages, but I just wanted to get an idea of how much work you then had to do, how many nuclei and things and follicles or oocytes that you looked at, and that would be wonderful. So if you address that afterwards, I fully commend you for the candidate here, for candidate of biological sciences, 1.5.23 in developmental biology and embryology, and paragraphs 9 and 11 have not been violated. Thank you very much.
Спасибо большое, Джанна. Асya Генриковна, вы можете ответить на прозвучавшие вопросы.
Профессор Джанна Бриджер, спасибо большое за высокую
оценку. Jana Bridger, thank you very much for high evaluation of my work. And you are interested in the fact how the figure of 86% of oocyte was obtained. All in all, 298 oocytes were analyzed according to different sections of chicken ovary, and I used 3D model. 244 out of 284 oocytes contained nucleolus. Thus, I received 86%. As for statistical data, for 95% level of relevance, the range of average figure of oocytes with nucleoli is 85 5.5 plus minus 2.4%. It proves that if we take another section for our analysis, 95% will be the part of this interval. If you are interested in the functioning of nucleolus and finding it and obtaining it, how many oocytes were in our analysis, I can't answer you right now because I can study this data further, but it was conducted on different samples, both mature and immature chickens, juvenile chickens, 95 samples, and the result was the same. And nucleolus functions at the stage of early diplotellus, and then it dissipates. The pattern of nucleolus functioning is the same, and correspondingly, it is useless focusing on the figures. I was interested in the quality. I hope that I answered your questions. Jana, are you satisfied with the answers?
Спасибо.
Yes, I am. Thank you.
Thank you very much. Distinguished colleagues, as our board members highlighted the main results of the research, I'm not going to repeat. I would like to make a generalization. Asya Genrikovna studies main general biological problems, the realization of hereditary information functioning of ribosomal genes in vertebrates, the problems of biological and embryological development. In connections to the problems, Asya received significant new results which could influence main scientific thoughts, and she showed the similarity in functioning of ribosomal genes in oocytes of mature and immature hens. She eliminated contradictions in earlier experimental data. Secondly, together with co-authors, she managed to find out and study functional gene 5S RNA in ribosomal repeats in vertebrates, and eukaryotes repetitions localized in different parts of genome was disapproved, and Asya systematized data about the existence of different strategies of different ribosomal genes, and these are main. All in all, I fully agree with my colleagues that the dissertation is accomplished at high international level, and she published article in prestigious journal Molecular and Biology, and conclusions are without doubt. The text of the dissertation is successfully structured and contains few misprints. I have a few questions. First of all, to prove the lack of amplifications of ribosomal genes at low rudimental level, as in lizard Podarcis sicula. I would like to see experiments on obtaining PC protein and the data they demonstrate the organization of ovary, gradients of maturing oocyte in it, and the characteristics of the material of the research. Small oocytes were investigated on separate sections. The dissertation on Asya Davidian on the topic functioning of the nucleolus agonized region in the growing oocytes conforms with the main requirements for the dissertation according to the order at St. Petersburg University, and our candidate deserves the degree of the candidate of biological sciences, specialization 1.5.23, developmental biology, embryology. Points 9 and 11 were not violated by our candidate. Elena, thank you very much for your detailed consideration and positive assessment of my dissertation. Concerning regarding the detection of the PCNA protein of the incorporation of EDU in the nuclei of small oocytes of Gallus G. domesticus, I think that such experiments would be useful. However, in my opinion, the presence of one or two nucleoli associated with a single nucleolus forming bivalent should indicate the absence of DNA amplification in chicken oocytes. And as for 3D AMIR program, in addition to the revealed gradients in the maturation of oocytes in the juvenile chicken ovary, 3D modeling helped to understand that we cannot analyze the presence or absence of the nucleolus in the oocyte without regard to the oocyte localization zone in the ovary volume. Gene's work was carried out on sections in which the zone of localization of the oocytes in the ovary was not determined, which could also affect the discrepancy without data. Thank you very much. Dear board members, are you satisfied with the answers of our candidate? Yes, I fully satisfied. I have a question. Are there volunteers from the hall to express their opinion? No volunteers. If there are no volunteers now, I would like to give the floor to the scientific supervisor, Elena Gaginskaya. Asya became a postgraduate student after she graduated from the university in 2015, and I would like to say that her topic was very new for her, because when she wrote her master's paper and bachelor's paper, Asya investigated the development of skull in axolotus, and becoming a postgraduate student and transition to this topic meant the transition to the new level of the research, and she was very enthusiastic about the research, about using new methods for this research. And she was very hardworking, and she received all the necessary theoretical knowledge and a great role and her a year and a half internship in laboratory of Jugolot, played a great role which helped her to become a professional. Carnegie Institute, Baltimore. I would like to note that and her communication with Mr. Domin. He is a very famous evolutionist, and it played a great role in her professional path. And Mr. Dmitriev also helped her a lot. And during these years, I understood that Asya is a great professional, a hardworking person. I want to say that these were very pleasant years, and she turned into serious researcher. And she has brilliant personal qualities. She is interested in science very much, especially is in developmental biology. She has got a very huge creative potential. She is very capable. She is enthusiastic, and brilliant education she received at the Department of Embryology helped her. And then her master's experience they cooperated with Malashichev Yegor on evolution of skull. They allow this experience allowed Asya to look at the science from a different angle, use a critical approach. I want to say that Asya is very responsible and very honest researcher. She's a critical thinker, and she's able to defend her position. It is a brilliant personal quality. And Asya is very kind person, sympathetic. She's able to work in team and is always ready to help. She's ready to participate in teamwork. She's always ready to help colleagues and students. She's a responsive person, not only responsible but also responsive person. And I'm sure that Asya is serious, completed and mature scientist in the sphere of molecular and evolutionary biology of development. I wish you every success and successful scientific career. Thank you very much. In the course of our meeting, and according to the live broadcast of our board meeting, there are no external questions. Dear colleagues, are there any, dear board members, are there Any questions, or those who are in the hall, do you want to express your opinion? No volunteers. The discussion and the acceptance of and the receiving of questions are finished. Dear board members and candidates, any remarks or questions or objections concerning the procedure of our meeting, as well as the remote interactive mode? Please, dear board members, answer in turn. No questions? Joanna, do you have any questions concerning technical parts of the procedure?
No, I don't.
No questions. And the next point of our agenda, voting. Dear colleagues, we have the opportunity, before we start our voting, to announce a break to discuss the defense results with the broadcast sound muted. Do you think we need such a break to discuss the results of the defense? No, we don't need. I agree with you. If a break is not required, we continue our meeting and turn to the voting. And the opinion of every board member is voiced independently and publicly. Anna? I'm for. Alexander? I'm for. And it is stated in my review, I'm sure the dissertation conforms with the requirements for the dissertations of Dmitry. I'm for. The dissertation conforms with all the requirements for the dissertations. And it was voiced in my review. Jana, what is your opinion? Are you for?
I am for.
Я тоже присоединяюсь.
And I join my colleagues, and I am for. And I am for. Counting of votes, and thus, distinguished colleagues, I announce that out of 5 board members who participated in the voting, 5 members voted for, no one voted against, no one abstained from vote. The decision on awarding Asya Davidian an academic degree of the candidate of biological sciences in scientific specialization 1523, developmental biology, embryology, is accepted, and the floor is given to our candidate. I am grateful to all board members. I appreciate that you had time to study my paper in detail. I would like to express my gratitude to the Elena Gaginskaya for her kind attitude, constant help, and help with articles. And I would like to remember my first scientific supervisor, Igor Malashevich, and thanks to him I appeared at the department. Елена Кошель, Svetlana Galkina, Alexander Demin, thank you very much for the introduction to the topic of the research. And without opening the gene of NOR5S RNA gene, without this discovery, my defense would be impossible. And Resource Center of St. Petersburg State University, Development of Molecular and Cellular Technologies. Thank you very much. Center Chromas, in particular Anton Radayev and ex-director Susa Fedinova, for their invaluable assistance in the implementation of experiments and, in general, an excellent working atmosphere. Express my gratitude to all professors and staff of the Department of Embryology of St. Petersburg University for their friendly attitudes, important advice, and knowledge I gained during my studies at the department. Many thanks to Anton Koma from the University of Cleveland for very valuable advice. Thanks to him, I got acquainted with laboratory of Sergei Dmitriev, Nadezhda Makarova. Thank you very much. I would like to thank Joseph Goll for the opportunity to intern at the Carnegie Institute, as well as to Deresheva Svetlana and many others for the help and available advice. Also, many thanks to the leading researcher of Vizor, Igor Belausov, for his support and assistance in mastering a number of computer programs. And I'm very grateful to my family, especially to my parents, who have always supported me and believed in me. This dissertation work was supported by RFBR grants, and the numbers are clearly seen in the slide. Asya Genrikovna, thank you very much. We congratulate you on this event, on this defense, successful defense. The meeting is announced closed. Thank you very much for participation. Please switch off the live broadcast. Goodbye.